Welcome back to the FARA interview
Hello.
series where we are taking you behind the scenes at the International Ataxia Research Conference here in DC.
All right. Would you introduce yourself to our audience at home? Tell them about your institution and
Yes, okay. So I'm Olivier Griso. I work in the IGBMC, it's an institute in Strasbourg in France. I work in the team of Hélène Puccio for like now 5 years. I'm a PhD student and I started as a master's student in her team. And we are basically, well, Hélène Puccio's work is really known in the community, but we are basically working in developing animals and cellular models of the disease and developing therapeutic strategies as well.
all right.
So, your talk this morning was very personal to me. And I may get emotional because I have put, and my community and the whole FA community has put a whole lot of effort into fundraising, and your work is funded with, it's called the Kyle Bryant Translational Research Award, but really it's a ton of people putting their heart and soul into fundraising. And so can you tell us about your research? And then I want you to let us know what it means to you that the FA community is funding the research.
First, I would like to thank you and thank FARA in general for this great support because it's a big help to develop this project that I really like. And I feel very delighted that you put your trust in things that I'm trying to develop, so it's a great pleasure. And about my project, so should I explain? Yeah. So it's basically we have developed a way to screen some molecules in a new kind of system because normally what is getting done in the companies is screening the molecules on very basic cells. So normally you take some skin fibroblasts, skin cells.
Give us a second. What is explain to the community? What is a fibroblast?
So a fibroblast is just a skin cell. So it's easy to take from patients because it's non-invasive. You just take a bit of skin and then you can culture them and grow them and keep them and study them. So it's easy to try to find some drugs in these cells, but as they are not really affected in FA, you don't get normally significant results because we know that the skin is not affected in FA. So what we propose with this model is to use from mouse directly the primary affected cell types, which are the neurons and particularly the sensory DRG neurons. So obviously,
That's the part of the brain, the dorsal root ganglia.
so it's not it's not in the brain. It's along the spinal
sure.
cord. Yeah, but it's the neurons that are primarily affected in FA. So obviously these neurons we cannot have them from the patients. So we have to either we generate them by means from skin fibroblasts. We have ways to change them and to grow them into these kind of cells. But our project is to take them from mouse. So we take them from mouse embryos. We harvest them on the mouse. We put them in culture, so we are able to grow them in some specific kind of plates, and then we will test some drugs in these cells. But the thing is that it's not really easy to culture them like that. There are some cells with really long projections, and if you want to screen some molecules, you have to grow them in specific supports that have really tiny wells, and normally in these tiny wells they don't really like to evolve. So the tricky thing was to establish a way to culture them in these plates and keep them, and then we can try to screen for many drugs. So we have been able to do that in what we call 96 well plates. There are plates in which you have 96 wells, so you can test one drug in every well, and it's quite new to be able to do that with these cells.
So, the thing I got from your talk, and obviously a lot of it was over my head, but one of the things I grabbed onto, and tell me if this is off base, but is that like normally we think of frataxin like something will die if it doesn't have frataxin, right? And so one of the points of your talk was that you discovered that there might be a way that we don't need frataxin, right? Is that...
Well, obviously we need frataxin, otherwise Friedreich Ataxia will not be so much a problem. But the thing is that we show that in these cells, which are supposed to be the primary affected cells, when we delete the gene, which means when we delete completely the gene from the cells, they don't die, at least they don't die straight away. It's very encouraging because we see they have troubles to function correctly. So I think which triggers what we know as symptoms in FA. but maybe they just don't die. So it means that they are still to be rescued. So it's very encouraging because maybe the patients, they have problems making these cells work, but they are not dead, you know.
Yeah.
Maybe. I mean,
But deletion of the gene in the survival of the cell suggests some kind of like a compensatory mechanism.
yeah,
almost.
yeah, of course. So there's a part of the project that will be to understand why and how they, because if we can investigate these mechanisms and try to target these mechanisms with some kind of drugs, we can maybe help them surviving even more. So yes, obviously they find ways to live without frataxin because frataxin is important, we know that, but it's very encouraging for the patients because apparently the cells are not lost, they are just, they have troubles to function. So if, for example, with the gene therapy, if we bring again the gene, maybe the cells will revive, come back to life somehow. They are not lost straight away, which is really encouraging.
It's amazing.
Yeah.
Yeah, but this is in this system, in these cells, we cannot apply everything to.
It's in a tray that has 96 holes with cells from a mouse that you've cultured specifically.
Yeah.
So there's a long way to go.
Yeah, there's a long way. So
Right.
you cannot always relate. Yeah,
But we've talked before about how we want to have these interesting ideas to push ideas into a pipeline. So eventually we can pull a therapy out of a pipeline. And all great discoveries in science start in the lab like this.
Yeah.
Yeah, and maybe this idea, maybe it does, but maybe it doesn't lead to a specific therapeutic intervention, but it informs. It's an idea that a finding that informs everything that everyone else does too. So, in that respect, can you comment on? I mean, there was lines at the microphones after your talk, and can you comment on how that feels like as a young investigator, having all these people being like, "Wait a minute."
Yeah. I was feeling very nervous first because there's a part of people that are here to congratulate you, to say it's nice, and the part that are not convinced. And so first, I'm really delighted that for the first time I'm able to talk about that to this community, because first it was only in the lab, and it's a project that I really wanted to develop, and today I was finally able to spread the word, let's say. So I'm really happy to have comments, bad or good, because every comment will make you go forward. Right.
As a young researcher coming into a conference like this, what's the value of coming to a conference like this?
It's a great opportunity for me to present my work. First, to have been selected to talk, it's already like a reward, because I've been working to develop something and someone is trusting me to discuss about my results. So it's like a reward, I would say.
Yeah.
And then I also get to get the comments in order to go forward,
to collaborate with others, to talk with other scientists.
Yeah, and to improve. I mean, every comment you get from someone, it makes you improve in your research.
Yeah, right before your talk, Ellen is the one who introduced you. She was the chair of that section of talks.
Yeah.
And she said, you know what, I told him this wasn't going to work.
Yeah.
There's no way you could do this.
Yeah.
And so that, I just think that's kind of cool. And she does obviously too think that's cool that this, there's so much to learn from what you did.
Yeah.
So the thing is, when you start research as a really young, when you finish your bachelor, you will go for a master and then PhD. In your mind, you're still a bit dreaming, and you want to help. I didn't know what to do. I didn't want to work specifically on FA. I didn't even know FA before I started working on it. But I wanted to do something that matters. That makes me think that I'm useful for something. A bit naive, because then when you go in research, you realize that it's not that easy, and there are many steps before you actually help someone. Right. So I had the project in the beginning to understand more of the physiology of these cells, understand how they developed in the absence of frataxin. It was more fundamental aspects. But at the same time, I wondered to myself, okay, how can I make this something that I feel useful to do? So I said, okay, maybe I will try to do this screening on these cells, because it was never done on these cells. And it's challenging because nobody ever bothered to try something like that. So I proposed the idea to Hélène, and as everyone, she said, don't waste your time doing that, because you're not gonna succeed. So I said, okay, but still I want to do it. So, and I got lucky, because finally I tried to do it. I optimized things, but it's working. It could be also not working at all, and finally it's working. So I'm really happy. And now she's happy as well, but I had to push a bit to get to that.
Yeah, I think that speaks to the value of ambition and like thinking way outside the box and pushing through when everyone else says, heck no, that's not going to work. So good on you for that.
Yeah. Thank you so much for your research. It's great for the community. We really do appreciate it.
Appreciate it as well.
All right. Be sure to tune in to the next in our continuing series of interviews with scientists and researchers. We're on the FARA Facebook page, FARA YouTube, FARA Twitter, and FARA LinkedIn. Until next time, we'll see you later.
Bye-bye.